Hello everyone, we are Paolo
and Benjamin,
co-first authors of a recent publication in the Journal of Cell Biology titled Automatic Detection of Spatio-Temporal Signaling Patterns in Cell Collectives. This work, coordinated by Maciej Dobrzyński, was conducted at the University of Bern in Switzerland, in collaboration with the University of Graz in Austria and Drexel University in Pennsylvania. Today, we are excited to share our research findings with you. This research was inspired by our intriguing observations of coordinated ERK kinase activity in epithelial cells. Similar patterns were observed in other studies, such as ERK waves during scale regeneration in fish and calcium waves following epithelial extrusion. These observations highlight the prevalence of collective signaling phenomena in nature, orchestrating self-organizing processes like homeostasis, morphogenesis, development, repair, and cancer. Recognizing the need of a computational tool to identify dynamic collective events in different systems, we developed ARCOS. ARCOS is a powerful tool that detects active or inactive cells in time-lapse movies and identifies clusters of synchronized activity among neighboring cells. It then tracks these collective events over time and extracts their statistics. To validate the ARCOS, we conduct experiments using optogenetics to generate synthetic collective events in living cells. By selectively illuminating specific cells in epithelial monolayer, we successfully demonstrated that ARCOS can correctly identify and track these events. Our tool also provides informative statistics, including the spaghetti plot, which visually represents the dynamics of collective events in space and time.
Through ARCOS, we explored the dynamics of collective ERK activity in wild-type epithelial cells and cells genetically engineered with oncogenic mutations in KRAS and PI3K, commonly associated with cancer. Surprisingly, both mutations resulted in an increased number and size of collective events. Furthermore, we discovered that these events were largely mediated by matrix metalloproteinases, as their inhibition significantly reduced the occurrence of such events. We also used it to detect calcium waves induced by extrusion and to quantify fascinating shimmering wave patterns produced by giant honeybees in response to predators. Additionally, ARCOS can be extended to three-dimensional analysis, as demonstrated in tracking ERK activity waves in mammary acinis in vitro. We provide analytical tools for comparing experiments, detailed documentation, and open source code for R and Python. To make it even more accessible, we've developed a plugin for the Napari image viewer that allows anyone without advanced coding skills to use ARCOS interactively.